Hello, everyone. Thanks for joining. I am fortunate enough to do the introduction for Kyle today. Dr. Kyle Cromer is our newest faculty member in the Department of Surgery here at UCSF and the Broad Stem Cell Institute. Before joining us, Kyle received his doctorate under Rick Lifton at Yale using next-generation sequencing to discover mutations driving cancer. He then complemented this work by moving into the field of gene therapy and genome editing, first in George Church's lab at Harvard and then in Matt Porteus's lab at Stanford. There, he developed a novel genome editing strategy to correct beta-thalassemia, which has been licensed to Graphite Bio for clinical development. Today, he will talk about his efforts to develop an analogous strategy to correct α-thalassemia. Thanks so much, Kyle, for putting together a presentation, and I and all of us are so looking forward to hearing it. General rules of engagement for this presentation format. We usually have our presenters speak for about a half an hour. This is our opportunity to develop communication and conversation and collaboration around α-thalassemia. So after the presentation, if you're comfortable, we ask that you turn on your cameras and feel free to ask questions in the chat so that we can present them to Kyle and to others on the call to learn and grow from our group here today. Thank you so much. And Kyle, I'll give you the floor.
Thank you, Billy. I'm delighted for the opportunity to present. I've really enjoyed being a part of these talks before, and the opportunity to contribute my efforts is really meaningful to me. And so today I'll be talking about developing gene therapy and genome editing strategies to treat α-thalassemia. This group is needs no introduction to the disease. It's the loss of HbA1 and 2 duplicated at some point in evolution. We have 2 copies of α-globin, and the mutation or deletion of up to all 4 copies can lead to clinically severe disease. All 4 copy losses known as α-thal major. And in terms of rare diseases, this is a fairly common variant. Up to 5% of the world's population harbors an α-thal variant. And it's very devastating disease to those that have it, but for carriers of 1 to up to 3 copies of α-globin deletions or mutation, it lends a selective advantage against malaria, which you could infer from the distribution map. And as far as a rare disease, the rates of this are thought to be rising due to population growth and human migration, as well as advances in treating milder forms of the disease. So in a healthy donor, we have 1 copy of beta-globin and 2 copies of alpha-globin known as HbA1 and 2. The loss of this leads to 2 problems. One of these is that too little alpha-globin leads to insufficient hemoglobin tetramers, leading to severe anemia. And then too much beta-globin alone without its alpha-globin binding partner form toxic dimers that can transport but don't release oxygen. And it's really the beta to alpha-globin imbalance, the degree of the imbalance that dictates the severity of disease. And so any correction strategy would hope to address this. And really, even though we have some great options for treating beta-thal and sickle cell disease, which I'll go into depth on, alpha-thal has been largely overlooked for gene therapy, mainly because it's historically lethal in utero. However, recently, intrauterine blood transfusions have been shown to allow survival to birth, at which point patients' disease can be managed as you would for beta-thal and sickle cell disease. There are curative solutions available for beta-thal, sickle cell disease, and alpha-thal, and these have been around since the 80s when an allogeneic means a healthy donor transplanted the bone marrow from a healthy donor into a patient, was able to cure sickle cell disease for the first time. And so the workflow of this is step 1, you need to clear the bone marrow niche to make room for the transplanted stem cells that have been isolated from the healthy donor and transplanted into the patient. And while this can be curative, this is majorly limited by the availability of matched donors. The majority of patients do not have an immunologically matched donor. In addition, the radio and chemotherapeutic conditioning regimens carry a very high morbidity. And then there's also, because even in the instance of matched donors, there's still the risk of immune rejection and graft versus host disease. As a solution to this, there's autologous HSCT, hematopoietic stem cell transplant. And this was first used in a trial for severe combined immunodeficiency, and the effects were successful, though temporary. But strategies are currently underway for treating sickle cell and beta thal using this autologous bone marrow transplant where the patient is their own donor. And the workflow for this looks like you remove the HSPCs by mobilizing them into the peripheral blood. You can then correct them either with a viral vector or through genome editing, which I'll describe in more detail. You then need to clear out the niche to really increase the engraftment rates, really clear out space in the bone marrow niche for these transplanted HSCs, and then transplant the corrected HSCs in the bone marrow and hopefully elicit a functional cure, which equates to transfusion independence. And this solves the problem of low availability of matched donors because the patient is their own donor. Therefore, this workflow could conceivably apply to all patients suffering from disease. And because we're using the patient's own cells, they carry a much lower risk of immune rejection and graft versus host disease. It still, however, does require this high morbidity myeloablation to clear the bone marrow niche. And right now, these strategies only exist for sickle cell disease and beta thal, and there are no equivalent therapies for alpha thal major, indicating a major unmet medical need. So these autologous HSCT workflows have reached the clinic for sickle cell disease and beta thal. And these can be approaches that we will model our alpha thal correction after, this autologous ex vivo correction and retransplantation of a patient's own cells. The first of these was developed by Bluebird Bio. This is a gene therapy, and so this uses a lentiviral vector that semi-randomly integrates into the genome, so it's not as targeted as the specificity of CRISPR genome editing, which I'll talk about in a minute. It's gene therapy to just deliver an expressed copy of the beta-globin to restore beta-globin production and therefore hemoglobin carrying capacity, oxygen carrying capacity to red blood cells. And this approach, in spite of the lack of site specificity, has been in the clinic for the longest period of time and has elicited long-term transfusion independence in 32 patients up to 2.5 years, and even some patients up to 6 years. A newer approach has been developed by CRISPR Therapeutics, and as the name of the company indicates, this is a genome editing, so it's very site specific. The idea would be that you would only modify one site in the genome, and this is to de-repress the fetal precursor to beta-globin, gamma-globin. So gamma-globin takes the place of beta-globin early in development, but it's turned off naturally in most patients. There are, however, point mutations that allow persistence of gamma-globin, and this can actually compensate for beta-thalassemia and sickle cell disease. And so they use the site specificity of CRISPR to go in and replicate some of these mutations in patients where gamma-globin has persisted. And over 70 patients have been dosed so far, and transfusion independence has been achieved in a number of them up to 3 months and even up to 1.5 years. And then the latest of these approaches is another genome editing CRISPR-based approach to correct the single SNP, the single one base pair typo that leads to sickle cell disease with the site specificity of a CRISPR and combined adeno-associated AAV approach. And the first patient was dosed this month. So really, these are some great promising correction strategies that could address conceivably all patients suffering from disease. And so due to the success of these therapies, we hypothesized in our work that we could use analogous strategies to correct alpha thal major ex vivo. The idea would be that, so these are postnatal therapies. You would use an intrauterine blood transfusion to allow the patient to survive to birth, and then you could deliver this therapy postnatally. This would, because the patient is their own donor, address the shortage of matched donors for allogeneic HSC transplant. And also because they're the patient's own cells, we would reduce the likelihood of immune rejection and graft versus host disease. All of this is a three-pronged, highly collaborative effort. We drafted up an R01 that we submitted to NIH that got funded. It's funded until December 2026. And so the three different arms of this, the entire goal of it is to develop an ex vivo autologous HSC transplant strategy for correction of alpha thal, taking a cue from what's been done for beta thal and sickle cell disease, using three separate non-overlapping strategies. In my lab, this work was done initially in Matt Porteus's lab, where I was a postdoc, and I'm continuing in my own lab. And this is using a CRISPR AAV-mediated genome editing approach to restore alpha-globin production. And this is similar to the graphite bio strategy. In AIM-2, being conducted at UCLA in Don Cohn's lab, they are restoring alpha-globin using lentiviral vectors, which is analogous to the bluebird strategy for beta-thal. And then in Tippy McKenzie's lab at UCSF, she's using a CRISPR screen to try to identify regions in the genome that could de-repress the embryonic precursor to alpha-globin, zeta-globin, which is analogous to the CRISPR therapeutic strategy. And really with these, any approach that's shown to be successful could then be compared head to head, and we'd be in a really unique position to compare these strategies in terms of engraftment rate, correction, frequency, and in a way that Don Cohn called it a bake-off. And so we're really excited about the prospect of this as a follow-up to the individual successes of these strategies. Really, the ultimate goal is to file an IND by the end of it for autologous ex vivo postnatal alpha-thal correction with our best vector or vectors. So for my aim, we're using genome editing. But before we edit the genome, we first had to read it. So I have to pay homage to the first human genome build, which was constructed 20 years ago. This was done in February 2001. It established what a normal human genome looks like. And for clinical genetics, this is really equivalent to the moon landing, to the point where I came into biology and research after 2001. And it's hard for me to even imagine how biology was conducted before this. And then really the next 10 to 15 years was a golden age of driver mutation discovery, of which I played a small part for endocrine tumors. And then what remains now is finding increasingly rare drivers of increasingly rare disease and parsing complex genetic traits. So we're left after the human genome and the mutation discovery, really discovering what are the typos in the DNA that cause heritable disease. Really, where do we go from here? And in 2012, the discovery of CRISPR gave us the answer to this. The Human Genome Project gave us the ability to read the human genome as if it were a Microsoft Word document. And CRISPR is specific enough that we can now think about editing this genome as Microsoft Word document using some of the same functions you would use on your computer. So this includes a find and cut function. So when I talk about CRISPR, the main enzyme that's been characterized is an enzyme called Cas9. This is just a protein that is able to grab onto an RNA sequence that we can give it, scan the genome. It's a 20 base pair sequence, which is expected to occur less than one time in a genome for a unique 20 base pair sequence. It can scan the genome, and if it finds an identical or near identical match, it can bind this sequence and then cleave it. And so this is like the control F function and then the cut function on your computer. And this cutting can disrupt specific genomic regions, ideally just one spot in the entire genome if it's specific enough. However, most disease is caused by broken genes. And so there are few instances where disease can be corrected by breaking more regions of the genome, though there are some spots such as the CRISPR therapeutic strategy. There's also the copy function. So this is cutting followed by the providing of a repair donor. So this is a region that, so we cut the DNA and then we provide a double-stranded DNA repair template, either packaged in a virus or as a long strand of DNA. And this can be exactly identical to the region that we cut with just one to a couple minor modifications that the cell's endogenous DNA repair mechanisms will use to copy this sequence that we've provided to change at even single base pair resolution. We can now do something similar with a new technology called base editors that you may have heard of. However, most disease is caused by loss of function mutations scattered throughout a gene. So correcting single base pairs, you exhaust the clinical potential of this at a certain point. What has more flexibility is the paste function. And so this is a way that we can do the same approach of Cas9 guided cutting of the genome. And then we provide a repair template that we have an integration sequence flanked by homology arms immediately adjacent to our cut site that wedges in this integration construct into the spot where we cut. And this is really powerful because this can be pasted anywhere that you can cut efficiently. And a therapeutic transgene placed at a safe harbor site could conceivably be a one-size-fits-all therapy for diseases caused by loss of function mutations scattered throughout. It also can be integrated really at the base pair level resolution. And so we can put these into place in the genome without a promoter so that it's driven by endogenous regulatory machinery to elicit something like red blood cell specific expression, which I'll go into in my own work. And really, CRISPR gives us the unprecedented control over biology to the point where we can apply engineering principles such as design, build, test cycles in living cells. And that's how I'm going to present my work. So first, the foundational work for my alpha-thal correction strategy was a paste function that we used myself and in Matt Porteous' lab at Stanford to correct beta-thalassemia. So beta-thalassemia is caused by loss of function mutations scattered throughout the beta-globin gene, HBB. As far as rare disease, it's fairly common, and it's especially deadly even with current clinical care. And so in the healthy instance, we have one copy of HBB and 2 copies of alpha-globin that can form a 2x2 adult hemoglobin tetramer. In the context of beta-thal, we have lost beta-globin production, which causes 2 problems. The too little beta-globin causes insufficient hemoglobin, leading to severe anemia, and then too much alpha-globin in the absence of its beta-globin binding partner causes accumulation of toxic aggregates. What we then did was, because we have 2 copies of alpha-globin, we asked whether we could cut site-specifically just one spot, one of these alpha-globin loci with Cas9. And indeed, we actually were able to cut HbA1 at extremely high efficiencies in a way that was specific and never cutting HbA2, then provide a repair template packaged in an AEV. So it's an adeno-associated virus that's especially good at delivering DNA templates that we provide into mammalian cells. And so what this allowed us to do is replace an entire coding region of HbA1 with our HBB transgene to restore hemoglobin tetramers and ultimately improve the beta-globin imbalance. Essentially, one editing event in this case addressed both clinical problems. Too little beta-globin was addressed by HBB knock-in, and too much alpha-globin was addressed by HbA knockout. So in adapting this approach for alpha-thal, we have an analogous 2 problems. And so we hypothesized that we could cut one of the copies of HBB with Cas9 and then provide a repair template to integrate alpha-globin at the HBB locus in order to restore our hemoglobin tetramer production and improve this beta-globin imbalance. Again, in the same way, we would be expecting to increase our alpha-globin levels and slightly decrease our beta-globin levels, addressing both of the clinical problems contributing to the disease. So in terms of these design, build, test cycles, first, we need to follow, this is essentially a roadmap for how do you go from nothing to a clinical correction vector. And so it is these iterative design, build, test cycles, the first of which is optimizing your find and cut function by identifying your best Cas9 guides. From there, we'll then identify the regions of the homology arms that best mediate our paste function into that region that we're cutting. And as you can imagine, you need to be able to find and cut efficiently before you could possibly paste efficiently. And then we're going to identify the best regulatory regions for mediating efficient editing and maximizing expression of our edited alleles. And then we'll test these clinical vectors in wild type and patient cells. And so the first step of this, we screen 22 guides in the HBB gene, targeting both intron 1 and intron 2, as well as the 3' UTR. And the idea is we didn't want to cut an exon because we didn't want to disrupt HBB expression unless we were also integrating an alpha-globin transgene. And so in collaboration with Bika Weinert at the Conklin lab at UCSF, we identified 3 guides, one of them in intron 1, 2 of them in intron 2, that we're able to find and cut at high frequency. And so then we can proceed to the next design, build, test cycle with these 2. And this also limits our search. We can discard the other 19 guides that didn't work. It also eliminates the possibility that we would go after this 3' UTR targeting strategy. And so using what we found from the previous approach, we can model this after our beta-thal correction, that paste function that we used to correct beta-thal in the Porteous lab. And so what we had done here was identify a site that could be cut at high efficiency in HbA1 and not HbA2 that resided in the 3' UTR. And then what we did is have a right homology arm that flanked the sequence immediately downstream of this cut site. But then because we wanted our HBB transgene to be driven by the HbA1 promoter and fully knock out a copy of HbA1, we asked whether we could efficiently integrate this vector by splitting off the left homology arm and allowing the cell's endogenous repair machinery to essentially replace the entire segment in the genome in between our homology arms with the entire region between our homology arms of our repair vector. And this was able to be, this was shown to be highly efficient. So modeling this alpha-thal correction strategy after a beta-thal approach, we designed constitutive GFP, so strong, always active promoters followed by a GFP fluorescent reporter to allow a rapid readout of our editing frequency, just to show how well of cutting and then pasting in any given sequence would be for the two different strategies. This shows our intron 1 donor. So we cut with guide 7 and then have these corresponding homology arms. And then we also have an intron 2 donor that can be paired with these intron 2 Cas9 guides. And so we use this workflow for our editing. So we use wild type primary HSPCs that have been CD34 purified, which is a marker of stemness. 2 days later, we will then take our Cas9 protein, combine it with our guide RNA, pre-complex them together, and then deliver them by electroporation into the cells, and then add on our AAV containing a repair template immediately afterwards. And 5 days later, analyze our integration frequency by just reading out GFP by flow cytometry. And we find that targeting intron 1 happens to be the most efficient compared to our intron 2 approach. And this also makes sense too. We're not splitting off the homology arm quite as far. And so it probably aids in the cell's ability to identify the regions of homology. And so in the third cycle, so now at each step, we're constraining our search using a guide RNA that targets intron 1 and then also intron 1 corresponding homology arms. And so then now what we want to do is, instead of having these constitutive GFP vectors, what we want to do is have promoterless alpha-globin integrating vectors that are also paired with a 2A cleavage peptide, which will link HbA expression with anything downstream and have a YFP fluorescent reporter downstream. Because these are promoterless, they would be expected to be off in HSCs. It's driven, we're integrating at the HBB locus. And so this is supposed to be off in HSCs and turn on as cells differentiate into red blood cells. And so because we paired this with a fluorescent reporter without a promoter, we should be able to watch YFP if it's doing what we think, this integration scheme. It integrates, and then any integrated cell, as it differentiates into a red blood cell and turns on the HBB promoter, we should expect to see fluorescence. And we can do this by performing an in vitro red blood cell differentiation protocol, a subset of which the cells should turn green. And so here we're just showing that all of the different conditions, our original GFP vector, our mock vector, and all of these cells edited with these 3 different vectors are able to efficiently differentiate into red blood cells to almost 100% in vitro. And this is just showing that our editing scheme is not disrupting fundamental red blood cell biology. And then this was really nice and in line with our hypothesis, is that the constitutive GFP vector is on at high levels and then kind of equilibrates at lower levels, but it's always on. Whereas all 3 of these other vectors start at essentially 0. And as the cells differentiate into red blood cells, we see all 3 of them turn on in terms of GFP fluorescence, giving us a really nice view of the kinetics of expression here and ensuring that all 3 vectors elicit red blood cell specific expression, which is what we would want for a clinical transgene. And as further confirmation of this, we can also take the cells at the very end of a red blood cell differentiation. This happened to be one that was less efficient. And so we have both cells that are non-red blood cells that are retaining some of the stem markers of HSPCs, such as CD45 and CD34. And then we have ones that have become erythrocytes that lose 34, lose 45, so we can subgate on these. And then they've gained CD71 and gained GPA. And so in our cells, even though we've bulk edited all of these cells, the ones that retain the HSC markers have no expression of YFP, whereas in the cells that have gained our RBC markers, we see very high levels and a very high frequency of edited red blood cells. So this is really nice confirmation, both this and the previous slide, that we've identified a red blood cell specific expression site to place our HbA transgene for correction. And we've done a number of replicates across multiple wild type HSPC donors. And we want to do this before we move to patient cells, of course. And so what we find is that both of the the regulatory regions, we don't see dramatic differences in their ability to elicit high editing frequencies or really to mediate high expression levels. They can actually be quite variable. But if we're going to limit the development of our clinical vectors, our favorite are the ones that have these untranslated regions flanking our cassette that correspond to the HBA1 gene, as well as those that correspond to HBB. And so we'll proceed to the next round with these two. And so what this looks like in terms of developing the clinical vectors is we can take these 2A YFP fluorescent reporter vectors and just remove the 2A YFP. So we have really the final clinical vectors here, potentially, as long as they correct disease in our patient cells. And so we followed a similar workflow, repeated this in a number of wild type donors. And we see really efficient RBC differentiation in vitro, again, showing that with these clinical vectors, we're not disrupting basic HSC or RBC biology. And then we're also able to edit these cells at quite high frequencies. Because we don't have a fluorescent readout, we can't use flow cytometry, so we have to use a technique enabled by droplet digital PCR, ddPCR, to quantify our percent edited alleles. And then we can use an equation to relate what the percent edited alleles would be to the percent of cells that have one or more editing events. And it's half or more with both these vectors, which is really promising as we move into patient cells. So the next step is editing patient cells. And so we're extremely fortunate to be working at UCSF, where they have a repository of these. And so the sources can be either from fetal liver, umbilical cord blood, or bone marrow aspirates taken at usually the 1-year follow-up. And so we establish a workflow. To our knowledge, it's the first time that this has been done with α-thal cells in terms of thawing, purifying 34s, editing, and then differentiating into red blood cells. So we follow a standard protocol. I don't need to go into the details necessarily, but we thaw and then we perform a red blood cell lysis to get rid of any residual red blood cells in these preps. And then we do a CD34 purification to isolate CD34 positive HSPCs, a marker of stemness. We count and plate these at a standard density, and then 3 days later, so we allow them to expand. They would be expected to double in this time. And then we edit them with both of the clinical vectors vetted in the wild type cells shown in the previous slide. And then we can begin our red blood cell differentiation 2 days later. At the early time point, we'll harvest genomic DNA to quantify our editing rates in patient cells. And then at the end of our red blood cell differentiation, we'll quantify the RBC differentiation capabilities, and then also harvest our red blood cell pellets for both mRNA and protein analysis to hopefully ensure that we've restored α-globin production to these patient cells in our in vitro model. And as of right now, we've successfully applied this approach to 1 fetal liver and 1 umbilical cord blood sample. And then with the help of a surgery resident in Tippy McKenzie's lab, just performed the same workflow with 2 bone marrow samples and edited these yesterday. So the results are pending. And so just to show some facts plots, this might not mean that much to most people, but we see very high viability just shown here. We sort out just single cells. And then what we can see is a very high frequency of cells that have been edited, patient cells that have been edited. This is an umbilical cord blood sample. They efficiently lose CD34 and lose 45. These are double negative cells, and then they become double positive for markers CD71 and GPA. And so because of this, the FACS plots would indicate that we should expect to see very red blood cells. And indeed, this is what we see. And so this is a really powerful in vitro assay now for correction of α-thalassemia. We can now model in vitro erythropoiesis in these cells from frozen banked samples. And so we've done the editing in 2 of the patient cells right now that we've analyzed. And so at each of the endpoints of these, we will harvest DNA, RNA, and protein. The DNA is used to measure editing rates by ddPCR. The mRNA, we want to look for expression of our integrated α-globin transgene, and we're establishing that assay right now. And then on the protein level, we want to use HPLC to establish that we've restored α-globin protein production to these cells. And so what we've seen so far in our initial analysis is that we see very efficient red blood cell differentiation compared to the mock and the edited samples. They all efficiently differentiate, showing that we're not disrupting RBC biology. And we're able to achieve clinically relevant targeting frequencies with both of these vectors, about 36 and 24% of cells expected to harbor 1 or more corrected alleles. The next step here that we're currently working on is ensuring that hemoglobin production is restored. And so what we can do is take these red blood cell pellets, apply them to an HPLC machine, which then gives us plots like this. It's essentially quantifying our levels, and we can measure this by the area under the curve for single globin chains, especially when our approach is expected to decrease production of beta-globin and increase production of alpha-globin. It's really important that we quantify this at the single globin chain level, in addition to our hemoglobin tetramer quantification. How much more 2 alpha, 2 beta adult hemoglobin tetramers are we making in our edited cells compared to the uncorrected ones? And so these results are pending. The future plans are testing this in vivo in a mouse model. And so the idea would be that we would edit patient-derived HSPCs that we've CD34 purified in the previous workflow, and then transplant these into immunodeficient mice that are able to accept human cells when they're transplanted. 16 weeks later, we'll harvest the bone marrow, and what we can do is by FACS, sort the lineages, and then in the individual lineages and the bulk samples, quantify our editing rates by ddPCR. And this will ensure that our edited cells are able to engraft up to 16 weeks, and then they can all reconstitute the hematopoietic lineages, indicating that it's not having some effect on HSC biology. We also, because these NSG mice, these immunodeficient mice, can't make mature human red blood cells, our best proxy for cells that were engrafted producing corrected red blood cells is to take the cells, the human HLA positive cells that engrafted, sort them out, and then do another in vitro red blood cell differentiation and perform the same downstream assays as shown on the previous slide. Then after this, we're going to perform secondary transplants. What this means is we just take some of the bone marrow harvested from our initial experiment, transplant this into another round of immunodeficient mice. And this is really the gold standard for, was editing able to be done efficiently and long-term repopulating capacity HSCs. And this is usually the final step of preclinical validation before proceeding to file an IND for correction of this. And that really is the ultimate goal here, is to develop an autologous HSCT alpha-thal correction strategy for phase 1 to 2 clinical trials. And we'll really do this with our best vector or vectors from the CRISPR AAV approach that I just described, CRISPR only one from the McKinsey lab, or the lentiviral approach developed by Don Cohn in our comparison study. And then really looking ahead to the future, the way that I see the field evolving a bit is more targeted myeloablation. We hopefully will replace the high morbidity chemo and radiotherapeutic conditioning, and this is being achieved by HSC specific antibodies, and this is currently in the clinic right now. And then there's also growing momentum for in vivo therapies. And instead of these ex vivo, even postnatal approaches, this could be done postnatally in vivo or even in utero, where you would deliver the gene therapy or genome editing vectors directly to HSCs where they sit in the body, which would bypass the need for both conditioning and this time and cost intensive hands-on ex vivo modification. So we're heavily invested in both of these approaches in the future. In closing, I'd like to thank all of my collaborators and everybody who's helped me with this work. Tippy McKenzie, Bruce Conklin, Don Cohn, Matt Porteous, particularly Georgia Gregory and Bika Weinert, and Hira Nakauchi, as well as my funding sources. And a real special thanks to the consortium. It's been a pleasure to be a part of this, to join a network of patients, clinicians, and researchers. It's been a pleasure presenting my research, and please reach out if you'd like to get involved. Thank you so much.
Fantastic, Kyle. This is the conversation and collaboration portion of our hour. And so if folks were comfortable to turn on their cameras, we'd be grateful. Feel free to put your questions in the chat or to flag a hand up, and you can ask your questions directly.
Hey, Kyle, I might jump in. It's Fraser right here. Good to see you.
Yeah, great to see you.
Yeah, you too. I know it's cloudy up there at that north end of the peninsula there. So pulling like really nice work, really nice evolution. I was wondering just a general, if you could comment on the, what you see as the primary challenges moving to the in vivo approach. I mean, clearly it would be highly advantageous to be able to do this in vivo as a therapeutic rather than the, the costly, ex vivo approach. What do you think are the primary barriers there?
Yeah, thanks for your question. I'm glad to have the opportunity to talk more at length of this. This approach where it's that paste function, we have the tools, it's really about delivering the tools. It becomes a delivery problem. All of this ex vivo correction, we solve the delivery problem by pulling cells out. Never are cells easier to modify than when they're sitting right in front of you.
Yeah.
It then becomes a delivery problem. It's getting it past the liver if you're trying to target your HSCs. And then, really for my approach compared to the lentiviral approach and the CRISPR only approach by the Tippy McKenzie lab, it's going to be really tough to adapt that to in vivo. That's not a low hanging fruit because Cas9, the enzyme and the guide RNA alone is usually all you could fit into a delivery vector. And so then to also provide a second delivery vector to provide the repair template is really, really limiting right now. And so I think my favorite approaches are these that could use a Cas9 enzyme and guide alone to correct, either by breaking regions that would de-repress some of the precursors, analogous to the CRISPR therapeutics approach. I really like the base editor technology, and I'm currently trying to get that into my lab as another tool that would be template free. But then there's also all of the immunogenic challenges of delivering these vectors, and if you restore, in your work where in the presentations I've seen too, where you deliver an enzyme or a protein that's not been seen by the body before, aren't you going to be presenting that on the cell surface and maybe clearing out the cells that you just edited? So I didn't spend too much time talking about it because there are a ton of problems, but there's a ton of potential too. So it gives the field something to shoot for.
Yeah, great. Thank you so much. That's a great overview. Thanks.
And Mark Walters put in the chat a question regarding whether or not you've created α-thal HUDEP2 cells and complete the editing in this model that can express the adult globin program.
That's a great question. So Tippy McKenzie's lab actually does have the most common double HBA1 and HBA2 deletion line. It's called the SEA, Southeast Asia deletion. And so we have heterozygous and homozygous deleted HUDEP2 cells that are also a really nice model for in vitro erythropoiesis. We did try to edit them, but whenever you move to a different cell type, you need to really optimize all the different variables. And so, in the wild type cells, in the best case scenario, we're getting, upwards of 65% of cells targeted. We were only getting about 5% of HUDEP2s. And so it felt like we would have to spend extended time really optimizing the electroporation, the AAV dose and everything to efficiently edit them before they'd be a really nice model for us. But we're very interested in doing that. And, there's also interest from our end in creating a HUDEP2 constant spring mutation. There is a single point mutation that contributes to α-thal disease. And so I think that would be a really great opportunity to model correction of this with base editors or site-specific editing techniques.
Thanks, Kyle. Was the AAV vector toxic to the cells, or was it some other problem?
We see a little bit of that, but I think that it's more the cells, the viability wasn't too bad. I think it was, we just used the electroporation protocol that we use for wild type cells, and that can make all the difference, from going from low efficiency to high efficiency. And I think it would just take a little bit of time to optimize.
Okay, thanks.
Yep.
And Citra Matar has her hand up. Would you like to ask your question, Citra?
Hi, good morning, everyone, or good evening, and thanks. I'm sorry I can't switch my video on at the moment. I'm very impressed with your work, Carl. I really look forward to the results, especially in the translational aspects of your work. Now, looking forward, how do you anticipate that we can make this kind of, molecular therapy available to those who need it most? Yeah. And that would be people who have limited access to healthcare, because as you know, as everyone knows here, the cost of this going forward is going to be pretty prohibitive. So what can you see going forward that we need to do now to make sure that this is affordable eventually? Thanks.
Yeah, I really like that question. And I think for all of the gene therapy, genome editing space, developing these really expensive therapies. I mean, there are therapies that have been approved in Europe that there have been no buyers because it's just prohibitive. And I think really as a guidepost, we need to make this as cheap as possible. It's really the number one reason why I'm so interested in vivo therapies. And in spite of some of the issues I just talked about, I think that's the way that we make them cheap. We do need to first figure out if you could change any region of the genome, what would you change and in what cell type? So the ex vivo is a really nice model, and it's a really good starting point for testing what that genetic correction does in the limited number of patients that could afford these therapies. And then what we can do as the delivery, because the in vivo therapies it's really a delivery problem. As those delivery vectors improve, evolve, we can adapt all of these ex vivo correction strategies to be packaged in vivo delivery vectors. And they could conceivably be delivered without myeloablation, really even outside of a hospital as a single injection. And that, to me, that's a guidepost of where the field needs to go. But it might be several decades before we really get there. But as long as I'm moving in that direction, I think I'm going to the right place, because especially the hemoglobinopathies primarily affect the developing world. We have to make it cheap if we're going to make a dent in the number of patients that are suffering from this. Yeah, thanks for your question.
Thank you very much. Thank you.
And Kyle, Pinyafat Picam had asked, how does the length of your donor template, HBB, affect the repair efficiency?
Yes, it does. The general rule of thumb is the larger your integration cassette that you're wedging in, the less efficient it is. And we do see that. But there are other factors that can still allow you to achieve high editing frequency. If your guide, your find and cut function is really efficient. And then it's kind of locus dependent as to how much of a hit you take in terms of your editing efficiency for integrating a large vector. But really that sickle cell approach where they're correcting just one SNP, it's just one change and the donor template is identical except for that one piece. It's really easy for the cell's endogenous DNA repair mechanisms to find the homology regions and just repair that one spot. And so, yeah, that's a general rule of thumb for these large integration. But there is technology that's about to be published coming from the Porteous lab that can bias cells towards homologous recombination away from just the simple break repair. And so that can be a way to compensate and really to promote the cell to go towards that pathway.
I had a question. In the start, you talked about Bluebird and their experience, and you shared that currently 32 patients are transfusion independent after 2 years and several after 6 years. And was that since their gene therapy, or had some lost transfusion independence? And I guess towards the end of your presentation, you talked about the second transplant of the mouse model being the gold standard of moving to human. So is that kind of to address or mitigate the concern of loss of editing?
Yeah, I mean, so any modification you do to cells, especially the ones that are most stem-like, there's the concern that you're reducing their repopulation potential. I don't know why there's that discrepancy between, I think it was what you're probably indicating, I think it was like 70 patients dosed, 20 or 30 that showed really nice responsiveness. I don't know what the story is on the other ones, whether, if I had to guess, I would bet that it was low engraftment frequency of edited cells. That's such a barrier right now for efficacy. I mean, we really need to get about 25% corrected cells into the bone marrow to achieve that transfusion independence. And anything much less than that, it could be that the cells stick around. It's just that maybe you didn't fully clear out the stem cell niche, or that maybe your editing was in some way, your modification was in some way toxic to your HSCs. And there's only so much, in 32 weeks that we can say in a mouse model. It really is that long-term follow-up that's the true standard.
Is the goal for this editing, because you said, I'll just continue asking questions. Oh, Citra has another question also, but I saw you saw like 25% efficiency or up to 50% efficiency. And if we're talking about an alpha-thal major where there's a 4-gene deletion, is it then moving them from α-thal major to hemoglobin H, or from α-thal major to a carrier status, where carrier status has less health implications than hemoglobin H? So you may be moving from like a severe chronic disease to a milder chronic disease. Is that a correct interpretation?
Yeah, that's exactly right. I think, because a healthy person has those 4 copies, there's the idea at some point. Right now we're editing one spot in the genome, but you could conceivably in the future stack some of these edits. Maybe we're just getting them to carrier status, but say if the Tippy-McKenzie approach was also working, where you can reactivate embryonic zeta-globin, if there was a way to do that safely and introduce 2 changes at 2 separate sites in the genome, that would be a way of really amplifying your efficacy. You could conceivably have cells that have multiple copies of now something that can compensate. And so that's another area. I didn't get a chance to mention that until right now, but I'm extremely interested in multiplexing editing as well. Instead of just there's a limit to how much you can change by just making a single modification. And so I think that's also another way to really improve the efficacy of this.
And it looks like Mark in the comments had talked a little bit about the long-term clinical implications as far as he had said. And Mark, feel free to speak for yourself, but I think that the target editing in α-thal major might be a bit higher because the hemoglobin H binds no oxygen. So you will need to hit a higher hemoglobin for transfusion independence.
I'm sure you've heard this before, Kyle.
I think the bar for correction is pretty high, but it's something to aim for.
Yeah, I don't think you'll know. I think 25% is fine to put in the IND. You might get some feedback about that.
Yeah.
You might consider putting together a clinical advisory panel to advise you on that, but I think 25% is a good place to start. So I'm not being critical.
Yeah, of course. Thanks so much.
Yeah.
Yeah, and in our experience, with our registry, we have patients whose partners are hemoglobin H and they're silent, and they're carriers with a 2-gene deletion, and pursuing IVF, for example, with PGT, and hoping to have a child that doesn't have α-thal major. And I've worked with families in justifying the transfer of a hemoglobin H embryo, and their experience of their partner having hemoglobin H and that being an acceptable chronic disease for them, still very different from α-thal major. Yeah,
totally.
Ava is on the call. She's a part of the Bake Off. I don't know, Ava, if this stimulated anything in your mind, or if you'd like to say a piece about what you're doing, not to put you on the spot.
Well, before she turns on, I think that they submitted an abstract that was accepted for presentation at a conference, if I'm not mistaken. So this work will really be out there in the public. They've made great headway in Don Cohn's group with the lentiviral approach. And that's also one where you could integrate multiple copies of your lentiviral cassette. And that might, in the bake-off, perhaps be a more effective way than just a single modification like what we're doing. So I'm not just rooting for my approach, I'm rooting for any of them.
It seems like if you're targeting beta-globin, HBB, we're not concerned that both alleles would be targeted and suddenly you've converted α-thal major to a beta-thal major.
That's it. So it's something I didn't mention, but it's certainly a concern if we're hitting editing rates that would lead us to believe that we're making a substantial number of biallelic edits. There's always a way that we can titrate it down a bit with, reduce your amount of AAV. Is just a really nice way to lessen any sort of even mild AAV associated toxicity and drop down your editing rates. We can do some clonal analysis to quantify our mono and biallelic editing frequencies to really ensure that, that would be counter to our aims if we are, curing alpha-thal by creating beta-thal. And we don't have the luxury, like with that beta-thal approach, to have a second locus that we can just always target and leave the other one intact. So yes, of course, a concern.
Hi, Sevi. She just got on camera, one of our stars. Was there ever a thought to target β-globin since it's not a locus that's expressed postnatally? And if it's exclusively a postnatal intervention, using that as the site for editing?
So it is helpful in the way that you just said, we wouldn't risk causing beta-thal in our correction of it, but we would also have to have some methodology for reactivating it as well, since it's silenced. And so, that is some way that you could stack the edits. I really think that multiplex editing, even in the ex vivo space, if you really could make any changes to the genome, you want to change multiple spots at once. And that could be a really great way to knock into the β-globin locus, use even a base editor to maybe reactivate if we find something in this CRISPR screen, to just combine strategies.
Well, I see folks here from Cooley's Anemia Foundation also, and just as a mild advertisement, I know that there will be some presentations around α-thalassemia editing and the disease focus day at the New York Academy of Sciences meeting in October. So those that may be interested in attending in person, it's in the great city of New York, or I'm not sure if there will be a virtual option, but this subject is near and dear to many of our hearts, and it will be, we've had a great collaboration with the Cooley's Anemia Foundation, and we want to promote their efforts and work they're doing too.
Mm-hmm.
Any other questions for Kyle this evening? Oh, thank you, Val, for putting it in the chat. And just as a reminder for you out in the world, providers and patients alike, our registry has been a valuable resource for information about families with α-thalassemia major, and also an opportunity to engage patients in contributing these precious specimens for the work that Kyle's doing. And we've been able to consent families under a different protocol to coordinate those samples that are so essential for this work. And so if you have a patient confronted with a prenatal diagnosis, continuing and wanting to contribute samples at birth or at other times and procedures in their pregnancy, please do reach out to us, and we'd be happy to speak with them about that opportunity also to forward this work. And we are organizing for our next presentation. You will see in your emails the subject and dates, which is yet to be set, but we do plan to continue these conversations and presentations iteratively. So we will see you all soon in the fall, prior to our Thanksgiving holiday, I assume, in the US. And thanks everyone for joining. I see Anthea all the way from Australia and folks from all over the world, Thailand, and what an awesome experience to get to see your faces and become global friends in this network. So we're so grateful for your time and interest and for joining us. Have a wonderful evening, everyone.
Thanks, everyone.
Thank you so much, Kyle. Awesome talk.
Yeah, of course.